We have a range of faculty members and students who will give you a glimpse into science and life at Michigan State. Their stories and experiences are amazing, and we cannot wait to hear from them. To get started, I'd like to welcome Neil Hammer, Assistant Professor in the Department of Microbiology and Molecular Genetics.
So the title is You Are What You Eat, Understanding Nutritional Requirements of Bacterial Infection. And by way of an outline, I hope, I'll be able to present what I'm going to discuss today. So first I'm going to talk about the pathogen that my group is interested in, Staphylococcus aureus, call it the original superbug, and I'll talk about what that means here in a second. We're going to discuss the nutritional requirements of bacterial infections. So what do bacteria eat while they're colonizing us? And I'm going to discuss an exciting discovery that describes the first, or a Staph aureus nutrient sulfur acquisition system. And I should mention that, this work isn't published, so you'll be one of only a handful of people that knows that this system exists, which I think is exciting. We're going to talk about how nutrient sulfur acquisition shapes interspecies competition. I'll conclude, talk about some really interesting future directions, and then I'll be more than happy to take any questions at the end of the talk. So let's talk about Staph aureus. Many of you probably know what Staph aureus is, but it is a gram-positive coccoid bacterium. This is a sciencey way of saying that it's a spherical shaped bacteria that has a really thick cell wall. And one of the reasons why we study it is because it is the most common cause of bacterial infection in the clinic. So, and in keeping with that, it causes a majority of the skin and soft tissue infections, endocarditis, septic arthritis, and osteomyelitis. And what this means is that Staph aureus is able to colonize and infect nearly every organ within the human host, right? And not only is it able to colonize there, it also causes a vast amount of disease within these locations, which means that the organism has developed ways or evolved ways to live with its human host. Now, something you might not know about Staph aureus is that about 30% of the people, 30% of the human population are innocuously colonized with Staph aureus on their skin or upper respiratory tract. So what this means in practical sense is about 30 to 35 people in the Zoom call are colonized with Staph aureus on their skin or within their nose and don't even know it. This is a pretty interesting lifestyle for Staph aureus, and it has some implication for the research that I'm going to discuss today, but we'll get into that towards the end of my talk. The other reason why we study Staph aureus, and the reason why you probably might be aware of it, is that there's widespread antimicrobial resistance that's associated with this bacterial pathogen. In fact, in terms of total morbidity and mortality associated with antibiotic resistant pathogen, Staphylococcus aureus far exceeds all bacterial pathogens. And in keeping with this, I'm going to show you some dated data here indicating that, or demonstrating the increase of what are called methicillin-resistant Staphylococcus aureus cases in the state of Michigan in the early 2000s. Now, the reason why you might know this pathogen is based on its acronym, which MRSA. So you probably heard of MRSA before, and that stands for methicillin-resistant Staphylococcus aureus. And in the early 2000s, Michigan and much of the United States was devastated by this increasing prevalence of resistant Staphylococcus aureus. Now, Michigan also had the dubious distinction of being the state where 9 out of the 12 so-called vancomycin-resistant Staphylococcus aureus were also isolated. And about a decade ago, this was a really serious infection where there was no treatment options available for folks that were infected with these vancomycin-resistant strains. I always like to show this map illustrating the MRSA cases in Michigan, and of course, we're in Ingham County. I realize that there are folks all across the country on the Zoom call, but the mitten here in the middle of the mitten, we have a modest rate of MRSA infection. But what this means overall, the widespread antimicrobial resistance limits our therapeutic options, necessitating development of new targets. So what I'm going to talk about today is understanding nutrient acquisition and how that might be a way to control bacterial infection. So what does that mean? What are the nutritional requirements? Well, life is all about chemistry, and some of the phenomenal chemistry professors that are panelists today will no doubt discuss this overall. But we can sort of divide life into elements, right? So we're all composed of atoms and molecules. And these atoms are really important to sustaining life, right? So we have bulk elements, macro elements, and trace elements. And as human beings, we obtain these elements to sustain our life through our diet for the most part. I'm sure that many of you took a multivitamin today. Those contain iron, zinc, copper, manganese, maybe molybdenum in some cases. But we need these elements to sustain life, right? And the same thing is true for Staph aureus. It's composed of essentially the same thing that we are. But instead of obtaining these elements from a diet, they attain it from us, right? So they are restricted to the elements and elemental sources within the host, right? So they can't go walk out and order McDonald's, for example. They can only eat what is inside us. And so this is a way in which perhaps we can control and preferably inhibit bacterial pathogen growth by perturbing its ability to acquire these various nutrients. So my lab has focused on the bulk element sulfur for a couple of reasons. One is based on some previous experiments that we performed that got us really excited about sulfur that I don't have time to get into. But the other reason why is it's really important, right? It's an essential element for life. And certainly this slide illustrates this, but why? Can we go a little bit deeper into why sulfur is important? It's really important for essentially 3 reasons. The first reason is metabolism. And what I mean by metabolism is essentially the catabolism of carbon sources to chemical energy in the form of ATP. And it turns out that that conversion and the chemical reactions that occur in metabolism, a lot of those chemical reactions require sulfur. The other reason is proteins, which are enzymes, are composed of these building blocks called amino acids. And 2 of those amino acids contain sulfur. And I'll provide examples of that here in a second. And the last reason is that sulfur is reactive, is highly redox active, right? So it senses and can respond to the redox environment of the redox potential of the environment, and can help by being an antioxidant and sort of sop up electrons that have gotten loose. Okay, so we decided to focus on sulfur. What is the buffet of sulfur sources available to Staph aureus when it infects us? So here's a list of some of the sulfur sources and sulfur-containing molecules that are present inside us that Staph aureus might come in contact with. Now, this is not an exhaustive list, so these are some select sulfur sources that I've decided to focus on here. But you can see here that there are 2 amino acids that I was referring to earlier, cysteine over here and methionine. We have some non-protogenic amino acids like homocysteine and taurine. And then what's really interesting about cysteine and then this molecule right here called glutathione, which I'll abbreviate as GSH, is that they have this free sulfur atom right here. And what this allows these molecules to do is act as a redox buffer. So what happens is, in the case of where oxidative stress occurs, free electrons can just happen. And what we like to do, what our bodies like to do, is keeps the reduced form of cysteine and glutathione in vast abundance. And so when these radical electrons happen, what occurs essentially is a disulfide bond between 2 of the reduced atoms forming these oxidized versions called cysteine or oxidized glutathione. And here's an example of the sulfur bridge occurring between essentially 2 cysteine atoms, or in the case of oxidized glutathione, reduced glutathione. Right? So does Staph actually eat these sulfur sources? And it turns out that for most of them, it does. Now we have a chemically defined medium where we add essentially the menu that Staph aureus can eat, but we can change the items on the menu. And specifically, we're going to change the sulfur source that's present in this medium. So first, we started with a medium that has 2 sulfur sources, methionine and sulfate. And it turns out Staph doesn't like these sulfur sources at all. But if we add some of these other sulfur sources that we discussed, glutathione, cysteine, oxidized cysteine, homocysteine, Staph aureus grows particularly well. Maybe not so much for homocysteine, but for cysteine, oxidized cysteine and glutathione, it grows really well. There is some conflict in the literature as to whether or not Staph aureus was using this particular sulfur-containing molecule, taurine, and our data is more consistent with the results that demonstrate that taurine is not a viable sulfur source for Staph aureus. So what I convinced you so far is that Staph aureus is able to utilize the reduced and oxidized versions of cysteine, right? Reduced cysteine, oxidized cysteine, reduced glutathione, oxidized glutathione, but it was unknown whether staph can actually use oxidized glutathione as a sulfur source. So the first new knowledge that my lab has been able to generate in terms of nutrient sulfur sources that Staph aureus utilizes is showing that Staph aureus is capable of using oxidized glutathione as a sulfur source, right? So here we have a buffet of sulfur sources, and so we need to focus in on one of these sulfur sources. And you can see that maybe I'm trending towards glutathione. So in terms of the flavors that sulfur come in, I'm a big fan of cookie dough ice cream. So we consider glutathione the cookie dough of sulfur sources. And that's for a couple of reasons. Again, some previous data that I don't have time to go into, but also there's a strong rationale for focusing on glutathione. And the first one is that, as I mentioned sort of previously, glutathione functions as a redox buffer in host cells and some bacteria, but importantly, not Staph aureus. Staph aureus does not synthesize glutathione. The other major point of rationale for why we're focusing on glutathione is that it's abundant in us, right? So we have reduced glutathione. It's an antioxidant, so it needs to be kept in high concentrations, and it's found in, for the chemists out there, in about millimolar concentrations within host cells. And not only is it found in host cells, but it's found in specialized host cells. So red blood cells are a significant reservoir of glutathione. And this is important as it pertains to Staph aureus, because Staph aureus makes toxins that lyse open red blood cells. And so it's been previously shown that Staph aureus lyses red blood cells to take up iron, to eat the iron, but it's also possible that Staph aureus might be lysing red blood cells to take up glutathione. And that's something that my lab is exploring going forward. We also know that other pathogens use host-derived glutathione as a source of nutrient sulfur. So there's precedent for bacterial pathogens eating the glutathione that's present in our bodies, right? And so with that, we developed a hypothesis that Staph aureus encodes reduced and oxidized glutathione acquisition systems that support nutrient sulfur acquisition during infection, right? And we know that this hypothesis is true, at least as it pertains to cysteine and cystine, because my group previously published about a year ago describing these transport systems. And so this hypothesis predicts that, because Staph aureus can grow in medium supplemented with reduced or oxidized glutathione, that it should have transporter proteins that are involved in acquiring glutathione, right? So how do we go about finding these transport proteins? So I'm a classically trained bacterial geneticist. Many of the chemists in the audience and our panelists probably know that. Probably realize that given my sort of brief introduction into chemical structures and how the chemistry works in terms of electrons moving around. I'm far more comfortable talking about genetics. So what does this mean overall? So I'm going to give you a little brief introduction into the central dogma of molecular biology, which is the basis for performing these genetic screens to identify these transport proteins, right? So basically, we know that DNA is the inheritable genetic material that provides information to make proteins, and those proteins function to provide a phenotype, right? So as an analogy, I have blue eyes, which means that my body is expressing a protein that performs a chemical reaction that produces blue pigment in my eyes, and the information for making that protein is encoded within my DNA sequence, right? And so we can do this, we can exploit this information and perform what are genetic screens. And one of the high throughput ways we can do this is by mutating Staph aureus cells over and over and over at different locations in its DNA, generating a mutant library. Now, I didn't do this. I was fortunate enough to have colleagues at the University of Nebraska Medical Center who generated this library, but essentially they're arrayed in what's called a 96-well plate. So each circle here in this 96-well plate represents a place where one distinct mutant is stored, and we can assay. So each one of these wells is essentially like a miniature test tube. But of course, we need more than 96. We need about 2,000 of these to exhaustively search or query the Staph aureus genome to find proteins that are responsible for the phenotype that we're interested in. And so this presents an interesting conundrum, right? This essentially becomes our haystack. So how do we find the needle within our haystack? Well, we exploit the phenotype. So what happens is each one of these wells, each one of these 2,000 wells, there's 20 96-well plates, 96 obviously mutants per plate, gives us about 2,000 mutants. We're going to assess how well they grow in medium supplemented with we use oxidized glutathione in this screen. So the first thing we do is we grow everybody in a sulfur-rich medium, and all the mutants will grow because there's so much sulfur in here, it doesn't matter where they get it, and they'll grow, as indicated by these brown dots. The next thing we'll do is we'll supplement those mutants into medium that's supplemented only with glutathione, right? So they can only grow if they can utilize oxidized glutathione as a source of sulfur. We incubate them and quantify growth in this spectrophotometer. And then we can obtain a growth curve where, again, over time, we're observing increased bacterial growth in the presence of glutathione. However, some mutants won't grow. And that's because they contain inactivating lesions within the DNA sequence that encodes the protein that is responsible for importing glutathione, right? So, of course, we found some mutants that didn't grow, or else, that would be the conclusion of my seminar today. But what was really interesting is that these mutants had some cool traits that I'll discuss here. So the first thing is, obviously, those mutants, when we isolated them, don't grow in medium supplemented with oxidized glutathione. So here are circles of wild type cells that grow well, but our mutant strains don't grow very well. They do, however, grow perfectly fine when we add cysteine or oxidized cysteine as a sulfur source, and they grow very well. But they also don't grow very well when we add reduced glutathione as a sulfur source. And I'll comment here for later consumption that this is micromolar levels of reduced glutathione. Again, these mutants don't grow well in reduced glutathione. The other interesting trait is when we map these mutants on the genome, they were all pretty close together. And in fact, a computer program told us that this DNA sequence likely encoded a transport system. Now, it didn't tell us exactly what the transport system was, but based on the phenotypes that we observed, my lab decided to name this the glutathione import system, or GIS, right? And that's largely because the program said, okay, this DNA sequence looks like what's called a nickel peptide transporter. But based on the phenotypes and the screen that we applied to find these mutants and to find these proteins, we decided to name this the glutathione import system. So here's the model by which we think that this system is working. So that has two proteins that act as a pore that shuttle glutathione or potentially catabolic products through the membrane. We have this A protein, GIS-A protein, that's responsible for generating the energy that supports transport into the cell. And then we also have this lipoprotein, which is on the membrane and potentially grabs either reduced or oxidized glutathione or their products. Now, we probably could have guessed that this transport system had something to do with glutathione, and that's largely based on this predicted protein or DNA sequence that encodes a protein that's called gamma-glutamyl transpeptidase, or glutathione. And that's been shown in other bacteria, and in fact, in humans, this family of enzymes degrades oxidized or reduced glutathione. And so the two big distinguishing features between these models, as you can probably tell, is the location of glutathione. And so my lab is trying to figure out where this GGT enzyme is located and figuring out whether these proteins function like the computer predicts that they do. Right. And so my hypothesis, we were only half right. So we were able to find an acquisition system that is important for Staph aureus utilization of reduced or oxidized glutathione. But using a virulence, using an infection, an animal model of infection, we were unable to determine whether this system had anything to do with virulence, right? So what we do is we take a wild type strain and infect mice with our wild type Staph aureus or with our mutant strains. And it turns out that the mice got sick regardless of which one we put in, which indicates that to us, at least in this particular infection model that we tested, that the GIS system doesn't play a role in pathogenesis. And so we were a little bummed out by that, but I think that there's two reasons why the system didn't display a virulence phenotype in mice. And the first reason is because if you remember our screen, we were looking at medium supplemented with micromolar levels of oxidized glutathione. But if you remember when I originally introduced the idea of glutathione as a source of nutrient sulfur, that it's present in millimolar concentration. So almost order of magnitude more glutathione. And so what happens is if we supplement medium with micromolar levels of glutathione, so way more glutathione, now the GIS mutant here shown in squares grows perfectly like wild type. And what that tells us is that there's probably another import system, another mechanism by which Staph aureus can acquire reduced glutathione during infection. So hopefully what I've convinced you so far is that the GIST system that we discovered, right, you're one of only a handful of people that know that this system exists in Staph aureus, so that's pretty exciting, is responsible for importing low concentrations of reduced and oxidized glutathione. And we also have evidence to suggest that Staph aureus encodes another glutathione transport system by demonstrating that the GIST mutants grow perfectly well when we increase the amount of reduced glutathione to physiological levels. So we started to wonder, well, okay, like how can we get into an idea, like why does Staph have this, right? It doesn't make, we saw no virulence phenotype associated with these mutants, so why would Staph have this thing? And so I developed a collaboration with Dr. Janali Ravi here on campus in pathobiology, and we just asked a simple question: do other pathogens have this GIST system? And we thought, oh yeah, well, certainly other pathogens would have it. But it turns out only Staphylococci, only species in the genus Staphylococci have this GIST system. This was really exciting to us, and it's not even all the Staphylococci, right? So here is our Staph aureus proteins, and we're looking at percent similarity. So the more green, the more likely it is that these other Staphylococci contain proteins that look like this GIST system. And what's really surprising to us was that this particular species, Staphylococcus epidermidis, looked like it doesn't have at least a full GIST system, which was really surprising to us. And the reason why this is important is in terms of the Staphylococci that doctors will see in the clinic, Staph aureus far outpaces many of the other species. But the second most common one is Staphylococcus epidermidis. And we were like, oh, this is interesting. Probably the second best well-known pathogen in this species of pathogens doesn't have this system. And what's even more intriguing is that Staph aureus and Staph epi are both considered commensals of the human skin microbiome. So they kind of live in the same place. Well, that was interesting to us. We have two pathogens here that live in the same place. One has a system and one doesn't. So is it possible that the reason why Staph aureus has the GIST system is to outcompete Staph epi on the skin? And so we tested that by simply taking, basically doing sort of an octagon type fight where we take equal amounts of Staph aureus and Staph epi, put them in a tube, supply either oxidized or reduced glutathione as the only sulfur source, and say, we'll incubate you for 24 hours. Who comes out? Who wins? Are there more cells of Staph aureus or Staph epi, right? And so if the ratio is 1, it was a draw. If it's greater than 1, that means that there's more Staph aureus. If it's less than 1, that means that there's more Staph epi. And you can see in the environments where we supplied oxidized or reduced glutathione, there's way more Staph aureus. In the environments that contain methionine, which is not a viable source for Staph aureus, but is a viable source for Staph epi, there's more Staph epi. Okay, that's cool. So that demonstrates that, yes, Staph aureus is much better at obtaining reduced and oxidized glutathione than Staph epi. But is that GIST dependent? So how do we determine that? Well, what we do is we set up the competition, but in this time, we genetically inactivate the GIST system. So this is a GIST mutant of Staph aureus, no longer has the ability, right, as I showed you before, to utilize these glutathione sulfur sources. And when we do that and ask it to compete with Staph epi, now we see that Staph epi wins. And what these results tell us is that the GIST system promotes Staph aureus competition against other Staphylococci that apparently do not encode this glutathione acquisition system. So hopefully what I've convinced you overall today and demonstrated is some really novel science that Staph aureus utilizes oxidized glutathione as a sulfur source. That utilization of reduced or oxidized glutathione in Staph aureus is dependent on this particular system, the GIST system, the glutathione import system that my lab has discovered, that there's likely another low affinity reduced or oxidized glutathione utilization system that's active in Staph aureus, though we don't know the identity of that yet. My lab is working hard to find it. And that GIST provides that the glutathione import system provides a competitive advantage for Staph aureus in a sulfur source dependent manner. Okay, so where do we go from here? There's still a lot of questions, like for example, this other glutathione acquisition system, but we also want to start exploring nutrient sulfur acquisition from the perspective of the host. So how does the host and how does bacterial infection affect this buffet of sulfur sources that's present? And what we're going to use is this innovative technology called imaging mass spec. Now, I had the benefit of training for my postdoctoral fellowship in the laboratory of the scientists who really pioneered using imaging mass spec in the context of bacterial infection. Now, I don't have time to go into the technology overall, but suffice to say that here is an infected kidney, and we can see that Staph aureus is present in these dark purple spots. And coinciding with those purple spots, what we can do is look for masses that are associated with bacterial infection. The way we do that is sort of with a laser. The laser ablates all the molecules at a specific location, and there's a detector that tells us, okay, these different masses are associated with the organ system at these locations. And the laser sort of rasters across the tissue slice, giving us information of the masses. And we can detect things like host proteins, calcium and manganese, but we can also detect sulfur sources. And this is the first sort of imaging mass spec experiment from my lab, where we took uninfected mice or infected mice. And what we saw in this particular tissue slice from a liver is that when the host was infected with Staph aureus, that it decreases the amount of glutathione in the liver. And we thought, oh, this is intuitive, right? Because glutathione transitions from reduced to oxidized forms, and due to the fact that infection causes inflammation in the form of an oxidative burst, that we'll see an increase in the amount of oxidized glutathione. So we nailed it. We'll get a science paper, no big deal at all. Turns out that's not what we see at all. We see also a decrease in the amount of oxidized glutathione. So it looks like the host, when it's infected with Staph aureus, says, we're going to take glutathione off the menu. We're going to take this nutrient solver source off the menu. And what that's called is nutritional immunity. And that's been established for other nutrients, but never for sulfur. And so we think we might have preliminary evidence describing a nutritional immunity in the form of sequestering sulfur from the bacterial infection. But we still have a long way to go to prove that. So here are the folks who do all the work. I'm entirely dependent on these people for doing the experiments. Everybody's lab kind of looks like this, right? We have to do everything by Zoom in the COVID era. But specifically, Josh Lensmeyer is a talented graduate student in my lab who performed much of this work. I also want to point out former research assistant, John Shook, who performed the oxidized glutathione screen. My collaborator, Sophia Lund, who performed a metabolomics screen that got us excited about sulfur to begin with. And I also mentioned Dr. Janani Ravi's work with the bioinformatics that showed us that maybe Staph epi doesn't have this glutathione system. Obviously, collaborators at Vanderbilt and Florida allow us to do some of this imaging mass spec work, the NIH for funding, the American Heart Association for funding, and MSU for providing the opportunity to do all this exciting work.
Hi, everyone. My name is Rachel Nasimbwa, and I'm from Uganda. As Angela said, I'm studying medical laboratory science, and this is my senior year. I have minors in global public health and health promotion. My interests, and that's also what I'm working on, is infectious diseases and immunology. My research mentor is Dr. Michael Bachman, and I work at the IQ building, and that's where our laboratory is. When I'm not doing research, I love hiking and biking and reading other books that are not science related. So I'm from Uganda, and I just put this map here for a visual representation. Why I chose to come to MSU was basically when I was doing my application process, I had one of the MSU recruiters come to Uganda to speak to us about different programs that are offered at MSU. And I was very attracted to the diversity and the couple of programs that I could pick from to be in the science field. I knew I wanted to be in science, but I wasn't very sure what I wanted to study. So having that opportunity to speak to somebody during my application process really helped me a lot. And I was also interested in the global connections because I know MSU is a very big school and there's a lot of people to meet. So I was interested in meeting people and networking. And those are the research opportunities that were offered, but also I got a full-ride scholarship to help me with my studies as well at MSU. So my interest in research is basically, as you can see here, my research is in HIV, and there's a lot of infectious diseases, but most of the burdens are around Africa or sub-Saharan Africa. As you can see, this is just a visual map of HIV, people living with HIV. And there's a high burden of disease in Africa with about 20.6 million people. And that usually contributes to the high morbidity and mortality rates, at least in most of these African countries. So my interest was to contribute to that and to be able to learn on how I can contribute with my education into resolving these challenges. So my research, we have this question of, is an HIV cure or vaccine possible? There's a lot of research that has been done. There's a lot of work being done about HIV, but there's still something missing. So we decided in our lab to contribute to these facts, and I'll share in a little bit. So HIV is still a public health concern because diagnosis in most countries, at least in low developed countries, diagnosis is still a challenge because of lack of the technology or the resources, and that usually exacerbates the challenges when it comes to treatment, the late diagnosis. And then for the therapy, there's medicines that can be used for treating the HIV, but there's a lot of side effects, toxicity, and a lot of challenges come with that to the patients. So this is just a visual structure of HIV, of how the HIV virus would look like. So we have the virus structure here, and it has a lot of genes and proteins that are encoded on it. So when the virus attacks the human body, it usually attacks the immune cells that would be fighting against it. And those are the T cells and the macrophages. And that's basically because they have this receptor, the CD4 receptor, and that's how the virus attaches and gets into the cell. So ideally the cell or each person that is infected, these cells should be fighting against that infection. But then HIV comes to basically attack these immune cells that would be fighting against it. So that just weakens the person's immune system, and they're not able to fight appropriately against this infection. And that's why the infection just keeps on increasing in intensity. So in understanding transmission patterns, a lot of studies have done cell-free virus, mediated viral transmission. And this is basically having the virus enter into somebody's body and then attaching to the cells. But there's a lot of research about cell-to-cell viral transmission, which is basically a person infected with HIV transfers their donor cells or the HIV-infected cells into a recipient. So this, when a person receives another person's cells, there should be an immune reaction that is elicited because these cells are different from your own body. But what's being seen is not a lot of immune reactions elicited when somebody gets these infected cells from another person. And our hypothesis is that these viral genes or the non-structured genes would probably be contributing to these cells not eliciting an immune response as they should. So here, looking again at the HIV genome, these are basically the structured genes highlighted here in blue. These are the ones that contribute to the structure of HIV, and the ones here in orange do contribute to the structure of HIV through the reverse transcriptase, and then the envelope genes as well, the ones shown here. However, there's a lot of non-structured genes, the non-structured genes here that do not contribute to the structure of HIV, but we're still investigating to see what their contribution is to the progression of HIV-infected cells in the body. So the genes that I'm studying are these 6: VIF, TAT, NEF, VPU, VPR, and REV. So these do not contribute to this HIV structure, but we need to understand what their contribution to HIV replication and infection rates is. And that's what my research is about. So what we're doing, that those HIV genes, the 6 non-structured genes that we're studying, we're using bioluminescence immune cells, and those are the macrophages and the T cells that I mentioned in the beginning, which HIV attacks. So that we're using these cells to transfect mice, and then would use Firefly bioluminescence to track cell survival and how these cells are able to continue replicating despite of them being different from what the cell is supposed to be having. And then we'll be using imaging to follow cell survival, as you can see here. And that will help us understand which non-structured genes are contributing to this cell survival. Is it one structured gene? Is it all of them? And what that contribution is. our long-term goal is to use this studies from the mouse models into the monkey models, just to see the progression, because this is the closest being to humans. So we want to see if what happens in monkeys would probably be the same thing happening in people as well. So we hope our research would be able to contribute to understanding transmission patterns, especially the cell-to-cell associated transmission, and how that progresses into the immune effects that we see in HIV-infected people. We also hope this information can be used in therapy to contribute to therapeutic methods that are being developed elsewhere, or other researchers in understanding how these non-structured genes contribute and how they can be used as a target. And that's the same thing for the vaccine. If they can be used as a target early on to stop HIV replication, or to stop the viral replication when somebody gets infected, that would be a very good contribution to developing both therapeutic and vaccine trials. So as I conclude, I'm a senior, so my interests are, I hope to continue with the doctorate in infectious diseases and immunology, and also continue using that to integrate into public health and how different strategies can be used to help reduce disease burden, especially in low-income countries. So this is just to say thank you to Dr. Bachman and my lab members, the Dean's Research Scholars Program for the continuous support, and the College of Natural Sciences for availing all those opportunities for us undergrads to contribute to research.
Okay, here we go. So my work is associated with how people learn chemistry and how we and research in that area. And the work that I'm going to talk about today is on this course for general chemistry. And the reason we're looking at these introductory courses is because they're the most critical to the retention and recruitment of STEM majors. We've known for a long time that these gateway courses are in fact incredibly important for students as they move through the university. So in 2013, MSU received an award from the Association of American Universities to redesign our STEM gateway courses. And when this happened, we essentially gathered together a huge group of people. We've got funding from AAU and from NSF. We are multi-institution, across the board biology, chemistry, and physics group. But what I'm going to talk about today is actually, I'm going to talk about how we transformed the large enrollment general chemistry courses, Chemistry 141, 142 at MSU. And this transformation, I'm just going to start with the end of the story. This transformation resulted in 740 more students a year passing the course with a C or better now than before the transformation. This is a lot of students. And I just want to let you know that these students are moving through the university. They do as well as or better in subsequent courses than students who are taking more selective general chemistry courses. So this is something that we're very proud of. It's the first and the last course that most people in college at MSU take in chemistry. And in a typical semester, over 4,000 students are enrolled. None of them are chem majors. The chem majors are in separate courses. The honors students are in separate courses. Lyman Briggs has separate courses, but everybody else is in this big course. And the question is, well, why do so many majors require chemistry? And according to us chemists, chemistry is the central science. This is a infographic from the International Union of Pure and Applied Chemistry. We believe we are at the center of all the sciences and everybody else is connected to us. And, you know, there's some truth to this. Most STEM majors require students to take chemistry, but not everybody is enthralled with chemistry, which just say no to chemicals, which is preposterous. We are physico-chemical systems. Chemistry is everywhere. We're made of chemicals. We can't not have chemicals. So, although chemistry is important to most sciences, but perhaps it's not the most beloved. So what we wanted to do was to design a new course with some goals in mind. We want the course to be a gateway to the future, not a gatekeeper. We don't want to sort students out. We want students to succeed. We want it to be built on what we know about teaching and learning. And we want it to be interesting, rewarding, relevant for students. And so this course that we developed is called Chemistry, Life, the Universe, and Everything. With apologies to Douglas Adams. If you're a Hitchhiker's Guide to the Galaxy fan, you'll know what I mean. And my colleague, Michael Klimkowski from the University of Colorado at Boulder, who's actually a molecular biologist, was a co-PI on this NSF grant that allowed us to move forward. So there's a large theory and evidence base about learning, science, and learning in general. And these are National Academy consensus reports on how people learn science. And here's discipline-based education research, which is the area of research that I am involved with, and I actually was on the committee that developed this consensus report. We look at how, at the intersection between disciplinary expertise and education. And so in general, we know some things. We know that experts' knowledge is organized differently than beginners. We know that students construct knowledge rather than receive it intact. So I may be lecturing away here, but I have no idea what you're thinking about. We know that context matters, and we know that culture matters. So I'm just going to talk a bit about each of these in the context of the CLUE curriculum. And so the first one is this idea of how we develop expertise. Obviously, we're not expecting students to become experts after taking, an introductory chemistry course, but we would like them to have more expert-like knowledge. So what we know is that experts' knowledge is organized into an underlying framework that reflects a deep understanding of the discipline. So what we have to do is we have to teach bits and pieces. We can't teach the whole discipline all at once. So we teach some bricks, and what we want is for these bricks to somehow self-assemble into a beautiful expert-like palace. All right. And it turns out that people need help with this. So in chemistry, what we have done and across the board is this idea of organizing learning around core ideas. It helps students develop more expert-like frameworks. And I'll try and give you some examples of this as we move along. Okay, but if we don't connect the fragments, the bricks, to the core ideas that allow us to develop more expert-like connected ideas, what happens is we just get a bigger pile of bricks. And so I want to give you an example here. Here's a passage that I'll read to you. The montylation of traxylene. It's very important that you learn about traxylene. Traxylene is a new form of Zianta. It's montylated in seristana. The seristanians crystallate large amounts of fevron, and then bracteate to quasi-traxylene, and so on and so on. Okay? Now, I'm going to ask you some questions. We're not going to use the quiz feature because you'll get the point in a minute. So what is traxylene? Well, obviously, traxylene is a new form of Zianta. Where is traxylene montylated? It's montylated in seristana. How is traxylene quaselled? We can go on and on. We can ask these questions, and students can answer these questions, right? Never mind that this is all complete nonsense. These words are made up, and yet in many introductory courses, this is what we do. We tend to ask questions that are fact-based. Students can regurgitate the jargon without understanding what it means. Okay, and you may think that I'm exaggerating, but I'm not. So here's an example of a traditional assessment from a former course. And if you look at it, it's all bricks, right? Start with the number of protons in the nucleus of a lithium atom, multiply it by the number of 3s electrons, and then add the number of unpaired electrons in oxygen, and so on and so on and so on. If you look at this question, okay, it is just a ton of bricks, right? There's no building of knowledge. There's no use of knowledge. Students can't come out of this they can only learn some facts. And the problem here is that facts are not enough. These fragments have to be connected to each other so that we can develop a more coherent knowledge structure underneath. All right. So we want to connect them. And the question is, how do we help the students connect their ideas? Well, we're going to help them use their knowledge. Okay. They're going to use their knowledge by using the scientific practices in which have been developed by the national academies again. Okay. These are the things that scientists do. Science is not just a body of knowledge. It's a set of practices, and these are things that we do. We ask questions, we develop and use models, we plan and carry out investigations, we can construct explanations, and so on and so on and so on. So if we can combine connecting to core ideas with scientific practices, then we can maybe help students. Okay, so for example, you may, if you've taken chemistry, fondly remember this kind of calculation, okay? There are many of these in a typical course. And in fact, there's no chemistry here. We could be talking about dragon's tears for all it matters, right? There's no chemistry. So instead, we can ask the same question. We can ask the same calculation, right? We're going to have students look at the structure. We're going to ask them to give a consequence of the results of the calculation and connect it to atomic and molecular structure by having them engage in argument from evidence. And they're going to use the model, develop and use the model to justify and explain a prediction with a core idea. Okay, so this is a very different kind of approach, right? It's more complex, more difficult, I think, but that's what we do. Now, the second thing that we know about teaching and learning is that students construct their own understanding. They do not, knowledge is not passed intact from the instructor to the student. Students have to do something with it. So I'm just going to show here a few stills from a video that was made by Chemistry and Engineering News. They came to make a video of our transformation of the Gen Chem course. And this is Amy Pollock, who's the director of Gen Chem. And she's teaching, she's lecturing, right? We have 350 students in this classroom. There's actually, we won't be able to change this. But these students can interact, right? When we redesigned the curriculum, we added graduate and undergraduate learning assistants who move around the lecture hall to help students as they interact. They're working on problems and answering clicker questions. So you can see students have a clicker right here. So there's a lot more interaction in the the lecture, and the students are engaged in lecture, in recitation, and in lab on constructing knowledge. All right. The third thing that I want to talk about is that context matters. All right. So one of the fundamental problems in education is that it's very difficult to transfer knowledge from one context to another. It turns out that when students say, and if you've ever taught, you will hear this, when students say, oh, I didn't learn that. I don't know about this. I didn't learn that in my previous course. They're not making it up. And it wasn't that they didn't learn it. It's that it's very difficult to transfer knowledge from one context to another. So, so here's an example of an activity that we have actually done a lot of work on, which is that the idea of how molecules interact, right? There are two big end ideas here. One is that molecules can form bonds, right? Which are represented here. And another one is that molecules can interact among themselves, right? That makes them stick together. And it's this interaction actually that's extremely difficult for students to understand, it turns out. But without this interaction, you know, enzymes can't bind substrates. Geckos can't walk up walls. DNA can't bind to itself and unbind without breaking bonds. All right, so here's an example. And what we want is for students to understand that this interaction is between molecules, not within. And yet what we found is that in a traditional chemistry course, what we see is that most students actually, when they're asked to draw a hydrogen bond, an interaction, draw a bond, okay? And in general, this is because they're not asked to use their knowledge. They, and we've replicated this in many, many instances now. It turns out that this is across the board. It's an international problem. It's certainly all across the United States. But the CLUE students actually do recognize where hydrogen bonds exist, that is between molecules, and make things stick together. Okay, so here's the most obvious case of where hydrogen bonding is important for many students. Most of our students are bio-sci, health science majors. And so most of them know that there's a hydrogen bond between the two molecules, and we can draw it. We ask them to draw it. We also ask them now to make an argument about which of these two sets of base pairs is strongest and would require the most energy to overcome. And so we're asking them, what's your claim? And how does your evidence support the claim? And when students do this, they have to think about what they're saying. They have to write an explanation or reasoning. So this is actually, again, engaging in argument from evidence, because you know, we're asking them to make a choice, and we're also asking them to bring in these core ideas. Okay, and the last thing that I'm going to mention is that culture matters. The culture of the course, the culture that you come from, the culture of the discipline, the culture of the university. All of these things make a difference. That is how people interact, how you're expected to interact, what are the goals, what are the explicit and what are the implicit goals. Okay, so what we know is that course culture can impact student attitudes, beliefs, and outcomes. And there are two groundbreaking studies talking about leaving, why undergraduates leave the sciences, and talking about leaving revisited, which was 20 years later. And unfortunately, there wasn't much change in what these researchers found, which was that the students who left the sciences, and this is a huge problem for us, because we're losing so much valuable talent. The students who leave the sciences were not leaving the sciences because they were underprepared or because they couldn't do it. They were leaving because, for many, many reasons, but many of them because of the culture, because of the competitive culture, the way that they thought they were expected to behave. So, for example, curving grades, that is where you say, well, you know, this many can get A's and this many are going to fail, is extremely discouraging to students. It enforces competition, and why, when you've entered a university, would you have a course where a fixed percent should fail a course? That doesn't make any sense. So the clue culture that we have, we don't curve grades or have arbitrary numbers. Students have opportunities to make mistakes and recover. So the kinds of things that we do, the activities that I showed you a few minutes ago, are the kinds of assessments that we do. And students have the opportunity to make mistakes. That's when you learn, right? If you're paralyzed by the thought that you can't make a mistake, you're not likely to expand your horizons very much. So we want students to understand this is what we're after. Course grades are determined by both exams and by what students do in these group assessments, recitation and homework. And we consider ourselves to be gardeners, not weeders. It's not our job to sort students. It's our job to help students learn. All right. And some of these cultural things are kind of very implicit. And we've just done a study where we were looking at what students thought was going on here. So we asked them, what are you assessed on in this course? And it was interesting when we compared students who were in a traditional course to a clue course. This is actually the organic clue course. And what we found was that students are quite aware of what's going on, right? So if they are expected to memorize and regurgitate, then that's what they know what's going on. So this is also part of the culture. So the impact on students, I want to just go back to it, is, you know, we've got more students passing the course. There actually was a little grade disparity at the beginning where males and females were not, females weren't doing as well as you would expect, and that's gone. They get better grades, but do they actually learn anything? I'm sure you're worried about that. I would be too. So we gave them a nationally normed exam and they do fine. And we, as I said before, they do just as well as the students who've taken more selective courses in the next course. We've also published a lot on this, which I don't have time to talk about. The course materials are available as open resources. So the MSU Libraries has this option. So we've saved students $5 million over the past, since 2015. So that's also something pretty good. And my take home message, I think, is if we want our goals, if our goal is to help students take that leap into the unknown, which I think is the purpose of education, it's not to memorize some facts, then we have to help them develop deep and useful understanding by providing opportunities for them to connect and use their knowledge. And we also need to bear in mind context and culture. I would like to thank a cast of thousands, in addition to the interdisciplinary group. This is my research group. Amy Pollock, who is the Gen Chem director, and Lynn Marie Posey, who was the first person to teach CLUE besides me, and all the grad students and teaching assistants. And with that, I will finish, and thank you for your attention.
Hi, everyone. My name is Brent Strong, and today I'm going to present a little bit about what research I've done at MSU, specifically regarding both randomized controlled trials and acute stroke. And here I have a picture of Austin Bradford Hill, who is actually one of the epidemiologists who first pioneered the use of the randomized controlled trial in medical science. And he also was one of the first to uncover the association between smoking and lung cancer. But before I get into my research, I do want to tell you a little bit about myself. So I'm originally from Saline, Michigan, and for those of you who are familiar, very close to Ann Arbor. So I grew up quite close to the University of Michigan, but I'm very happy I decided to come up to Michigan State. I'm a senior majoring in physiology, and then I also have a minor, much like Rachel, in global public health and epidemiology. And I'm very interested in epidemiology, and that's actually what I want to study for my career. And it's a good time to do it in light of the COVID-19 pandemic. My research focus has actually been on acute stroke. Even though I'm a member of the College of Natural Science, I've been working with Dr. Matthew Reeves in the College of Human Medicine. He's a chronic disease epidemiologist who studies the clinical epidemiology of acute stroke. And I've worked with him during my entire 4 years at Michigan State University, which has been an incredible opportunity. And then come September, I'll actually be attending graduate school at the University of Glasgow in Scotland on a Marshall Scholarship. So I'm really looking forward to that. I'll be doing a master's in statistics before I hopefully go on to a PhD in epidemiology. And as I said, my research focus has been mainly on stroke. And for those of you who aren't familiar with stroke, stroke is a brain disease. And there are 3 different types. The first one is an ischemic stroke, and it results from the blockage of a blood vessel in the brain by a clot. And the result is that certain portions of brain tissue that receive blood from that artery, they don't get enough oxygen or nutrients, and they actually die, forming what's called an infarct. And then there are 2 other types, intracerebral hemorrhage and subarachnoid hemorrhage, which result from the rupture of blood vessels, and the same thing occurs. There's not enough blood supply, and these areas of brain tissue die. And unfortunately, stroke is extremely disabling. It's one of the leading causes of death in the country and in the world. And it produces all sorts of different impairments. So people who have stroke are often unable to walk. They may have something called aphasia, where they're unable to talk or comprehend speech. And actually around 800,000 Americans every year suffer from a stroke. So issues associated with it are very widespread. And actually the treatment of stroke historically has been very difficult. The first effective treatment for stroke was actually demonstrated 25 years ago in 1995, so fairly recently, and it's the use of intravenous alteplase for specifically ischemic stroke to help reestablish that blood flow through that blocked blood vessel. And the way in which these treatments are evaluated, like intravenous alteplase, is through the use of randomized controlled trials. And I want to talk briefly about what a randomized controlled trial is. So it's fairly simple in its conception. Basically, you have a sample population of patients, and they're randomized, as depicted here, to one of 2 groups typically. In the first group, they receive some sort of new treatment. And in the past 10 years, one of the main focuses in stroke treatment and stroke epidemiology has been endovascular therapy. And that actually is when the doctor will go in through the femoral artery, which is a blood vessel in the thigh, and use a catheter to go up into the brain and mechanically, physically remove that clot. And then the second group, they're randomized to a control. So that's usually something like what the standard care is compared to that new treatment, or some sort of placebo. And then after a certain period of time, you assess an outcome. And typically in stroke trials, you're assessing functional disability. And that really comprises people's ability to walk, ability to live independently, complete activities of daily living. And it's measured with a scale called the modified Rankin scale. And that's actually the key point of most stroke treatment. It's not to reduce the amount of death, it's to reduce how disabled survivors are. Now, one of the key parts of the design of a randomized controlled trial is that you project how many patients you're going to actually include in the study. That's in the sample population. So that might be 500 patients that you plan to include, or 750 or 1,000. And it really depends on the trial. But the important thing is that you do that before the study commences. However, one thing that investigators often do is that they'll look at the data before you actually get 500 patients into your study. So you look at it when 250 patients are included. And this may result, based on the results of those analyses, in stopping the trial before it completes, before you actually get the 500 patients. And one of the key reasons this occurs is that you see the treatment is very beneficial compared to the control. Now, there's a variety of reasons why you would want to stop if you see that. The first is ethics. If the treatment's highly beneficial, and we know that stroke is extremely disabling, it's not really ethically defensible to continue giving people a controlled treatment that doesn't work as well as the active treatment. So there's strong ethical implications for stopping the trial. Another one is money. These trials are extremely expensive, especially in the United States. I've been working on one trial, and this was actually not a drug trial. It was just some sort of social intervention with a social worker that cost around $10,000 per patient. So they're extremely expensive. And these drug and device trials, like intravenous alteplase or endovascular therapy trials, are extremely expensive, much more than $10,000 per patient. So there's a lot of money that is actually saved by doing that. And of course, people who run these trials, they're highly invested in a positive outcome. They want to see something that benefits patients. So that's another reason to stop. However, there's some key issues involved with that. When you actually stop early for a variety of statistical reasons, before you get to that number that you plan to enroll, say it's 500, you're very likely to overestimate how effective the treatment actually is. And this is data taken from a real trial that tested the use of albumin, which is a very common blood protein. So infusing that into patients' blood who have ischemic stroke, so the stroke caused by a blood clot, versus the injection of saline, so just the salt solution that's not really going to have any clinical activity. It won't help patients at all. Now, if we look at it early on in the trial, when about 275 patients are enrolled, we see a huge effect size. Albumin looks really effective because on the y-axis here, the cumulative proportion, that's the proportion of patients that are not severely disabled after their stroke, at 3 months after their stroke specifically. And that number is 45% among albumin, patients who received albumin, and only 30% among patients who received control. That 15 percentage point difference is huge. It's even bigger than the percentage point difference from the trial of intravenous alteplase, which actually causes the blood clot to break up and restores the blood flow through that vessel. So that's very surprising. You would not expect albumin to be so effective. And if the actual people who are running this trial had stopped it early, they might have thought that albumin is 15 percentage points better than control. Well, as you can see, the difference between the two groups starts to go away the farther you go into the trial. After about 550 patients are enrolled, there's only a 7 percentage point difference. And that'd still be very significant. That'd be very clinically important. And if that was the true difference, then it would become a mainstay of stroke treatment. However, by the time you reach about 850 participants, which is how many they plan to enroll, you see that the effect sizes they're basically the same. There's really no effective albumin treatment. And other data backs that up. Albumin does not really help patients with ischemic stroke. This illustrates the key issue with stopping early. When you stop before you actually reach that planned number of patients, especially because you do an analysis, you see that the data shows that the treatment is beneficial, you're highly likely to overestimate how beneficial it is, or even think that a treatment that doesn't work does work and has a very large benefit. So what I've been doing with Dr. Reeves' research group is we've been looking at how common this is among stroke trials, what sort of things are implemented to help reduce bias, and then also evaluating how big the overestimation may be among stroke trials. And we found that this is actually very common among stroke trials, and it's associated with what is likely a slight overestimate in the benefit of certain treatments, like endovascular therapy, which I've already mentioned. And that paper is currently under review at a journal. And I've been participating in some other different research areas. One of the key ones is sex differences in stroke. So men and women, there are some very key differences between them when it comes to stroke. For example, women are more likely to present with atypical symptoms. If you think of a stroke, you often think of things like facial drooping, slurred speech, muscle weakness, those very, very typical stroke symptoms that are outlined in something like FAST. But women are more likely to present with atypical symptoms, which may reduce the time or increase the time it takes to recognize a stroke. Another key one is that women are more likely to have severe ischemic strokes. And they're also more likely to have strokes at older ages. So these strokes can be especially devastating in women. Now, with that in mind, one of the key issues, and I've already mentioned IV thrombolysis, which is basically IV alteplase, is the mainstay of stroke treatment. And back in like 2009, my boss published some data indicating that women are less likely to receive intravenous alteplase than men. And one of the projects I was able to work on that we ended up publishing in Neurology was looking at whether this sex difference continues. And obviously, if women are less likely to receive this very effective treatment for stroke, that's concerning because they have more severe strokes that are more disabling. And we pulled data from a bunch of different studies from all over the world. And we found that indeed, women are about 13% less likely to receive intravenous alteplase compared to men. Now, there's a variety of legitimate reasons this may occur. One of the reasons that people don't receive alteplase is because they have high blood pressure, because then you're at a high risk for complications. And there's some data indicate that women are more likely to have high blood pressure when they come to the hospital. But regardless, this is a very concerning number and indicates that women are less likely to receive this drug than men. Although we did find some evidence and some other studies seem to indicate that the difference is narrowing. So we hope that it continues to narrow and is hopefully eliminated in the future. And I've very much enjoyed my time at MSU conducting stroke research, and I'm looking forward to my studies at the University of Glasgow in statistics and hopefully collaborating with some of the great stroke research groups that work there, that work on things like intravenous alteplase and other stroke treatments. So I'm excited to continue my study of stroke when I get to the University of Glasgow. Thank you.
Yeah, so I'm Johannes Palonen. As Corey said, I'm a professor in the physics department here at MSU, and it's my pleasure to tell you all about work that we're doing in my group, the Laboratory for Hybrid Quantum Systems. We're in the BPS building. That's where I am this morning. There's a picture of what the lab looks like on the inside, and what my group works on, broadly speaking, is the development of new quantum technologies and in things like quantum computing, quantum sensing, the development of new quantum materials. And I'll tell you about all the exciting things that we have going on in those kinds of arenas. But no, Corey mentioned the cat, no good quantum science or quantum computing talk would be complete without a whole host of call outs to Schrodinger's cat. But just as a caveat, no cats were harmed in any of this quantum computing research. I'll mention also that I'm an associate director. Angela Wilson is the director of a new center we have here at MSU called MSUQ. It's a center for quantum computing science and engineering, and there's a whole host of faculty involved from a variety of different departments, all working on various aspects of quantum information science and quantum technology development. And I'll say a little bit about some of the collaborations that are going on at MSU broadly in that regard. So just to give you a sense of where we're going and kind of give you a primer on what kinds of things I hope you'll take away from this talk, I'll give an introduction, a crash course in quantum physics, quantum physics 101 to get us all started. Don't worry, there's not going to be any equations and stuff. I'll try to be pretty qualitative but hopefully you'll get a sense of kind of the counterintuitive nature of quantum mechanics and the quantum world and why it's like that. And it turns out that quantum world and the rules of quantum physics actually enable a whole host of exciting and emerging technologies in computing. So many of you have probably heard of quantum computing. It's all the rage these days. Quantum sensing, quantum materials development, and I'll tell you about all the things that we're what those are and why they're exciting. And then I'll highlight what kinds of work is being done at MSU in that direction. Both, I'll focus primarily on the work that's being done at my group, but call out the wonderful collaborations we have with a variety of faculty at MSUQ. Okay, so if your life is at all like mine, quantum physics and quantum computing is absolutely everywhere. But in seriousness, it is really everywhere. So you've probably, it's in popular culture. Lots of movies have aspects of quantum physics or the quantum world in it. But outside of popular culture, there's, lots of places that you can interface with quantum physics. So for example, if you play video games, there's this wonderful, those of you that are familiar with Minecraft, there's a mod for Minecraft called QCraft, which teaches the laws of quantum physics, but within the world of Minecraft. It's pretty fun. I don't play that, but I've been told by students that it's pretty wonderful. So if you're like me and you have little kids, you can start teaching them very, very early all the important things that they need to know in order to be up to speed on quantum physics. So this is a picture. It's an old picture now of my oldest son. This is when he was a baby, but we get him started early, and he had a midterm shortly after studying this book. There's a whole host of these X physics for babies, so like astrophysics for babies, quantum physics for babies. You can find them on Amazon from this one, but this author, they're really terrific. And if you look in any popular media outlet, so I've pulled an article that's a little bit old. It's a few years old now from The Economist, but you can look in Forbes, TechCrunch, whatever. You can find a whole host of articles and news pieces about quantum technologies and quantum computing, and to try to give a sense of what is going on. And it's an exciting time because we're really at a stage like this article title is alluding to here. Quantum technology is beginning to come into its own, where we can now start to envision a new revolution in computing and in sensing and in materials development that kind of utilizes these laws of esoteric, what used to be just academic people studying quantum physics for the sake of studying quantum physics. But now we've gotten to the stage as a species that we can actually start manipulating quantum objects and like making real technologies out of them. And that's one of the aspects that I find really exciting about our work. And in fact, if you want to like get hands-on experience with real quantum technologies, there's a whole host of quantum computing companies out there, and many of them have their quantum computers connected to the internet via cloud providers. So IBM Q has a website where you can go and you can actually you can learn how to run and write programs on their quantum computers that exist all over the world. Amazon Web Services now has Amazon Braket, which is their cloud provider for various kinds of quantum hardware. So you can actually go to these places and sign up for accounts and actually do use a real quantum computer. I'll mention their toy systems. They're relatively small. They don't have the full power of what will hopefully one day be an entire new paradigm for computing, but they are great for learning how to do things, starting to imagine what can happen. And I mentioned already, but Angela Wilson, myself, and Andrew Chrislieb in CMSE founded a new center here at MSU called MSUQ, and to try to kind of, help to drive and accelerate this kind of technology development among faculty here at MSU. Okay, so Quantum 101, here's the crash course. So quantum physics is essentially the framework that allows us to understand the microscopic world, it's a world that we don't have everyday experience with, and nor do we have good control over it. And so things that are microscopic and subject to quantum physics are things like atoms, electrons, nuclei. These things ultimately obey the laws of quantum physics. And these laws are very, very distinct from the laws of physics that describe the everyday world that we do have experience with. So like, baseballs, weather patterns, cats, those are all classical physics. You don't need quantum mechanics to understand how those things behave, but to understand how atoms behave and electrons and nuclei, you need quantum physics. Similarly, another kind of object that's a quantum object is a photon, so a particle of light, that's a quantum object. Things that are not quantum are cats. So I already mentioned that. Now, these kinds of particles and microscopic quantum objects obey this very, very different kind of law of physics, and it's counterintuitive. So, for example, I'm going to describe a couple of just two salient features of the quantum world. One is called superposition, and the other one is called entanglement. And you'll see that these are going to sound on the surface of things kind of like they're very strange and counterintuitive. But this kind of disconnect with our intuition really comes about because of this business that the quantum world is not something that we've ever built our intuitions on. We don't We don't handle electrons or atoms or nuclei or photons on a daily basis. And so our intuitions, you know, there's no need for them to be connected with the quantum world. And in fact, the language that we use from our everyday life to try to make sense of what's happening in the quantum world is actually pretty imprecise. And so that's what leads to some of this counterintuitive business. But, you know, the theories and the mathematics are very, very precise. And in that language, the quantum world is very well described, and that theory of quantum physics matches beautifully with the experiments that we can do. And it's actually the fact that works out so well that lets us try to do to build technologies based on this. So anyway, let me describe superposition. So superposition in our imperfect, imprecise classical language is, if you imagine you have a single object like an electron, and if it was in, say, the superposition of two of its states, what it means is you could imagine saying things like, you know, the electron is in one place and another simultaneously. Now that makes no sense classically, right? You can't have a cat be in one room and another at the same time. It has to be in one or the other. But this is, again, mostly an issue with language. But the electron, in a precise mathematical description, can be in this kind of strange superposition state of having many different properties, all that seem kind of impossible to hold at the same time, but yet it does. Now, entanglement is another aspect of the quantum world where you have, say, two objects. Here's a picture from the National Science Foundation of, I think these are supposed to be two electrons or two ions or something, where you have two of them, and they're entangled with one another. And what that means is that each of them loses their individual identity. So in an entangled state where you have multiple quantum objects, two at least, you can't really distinguish between one or the other independently. You have to talk about them intrinsically as a single object that has properties that can't be disentangled into two individual parts. So those are, again, that's again a process that we don't really have a good classical intuition for, but it is very precise in the quantum world. Okay, so why do we care about all of that? You know, this weird quantum world. Well, you know, there's the fundamental physics aspects that I already alluded to. So nature fundamentally speaks a quantum mechanical language. Here are these electrons and atoms and photons all talking to each other via this wonderful language of quantum physics. This is from the PhD comics, a movie that they made. So this kind of fundamental physics approach to try to really understand what's going on within nature at its fundamental levels is, these motivations are ones of exploration and discovery and trying to figure out what on earth is going on in the world that we live in, and how well can we understand it. But we've actually known about, as a species, we've known about quantum physics for a long time, over 100 years now. But we've only recently gotten to the point where we can actually start controlling and creating larger scale quantum objects, not necessarily just single electrons or atoms or just looking at what nature is doing, but actually engineering, say, here's an example of a chip that has superconducting circuits that form qubits, and I'll describe what those are in a minute. But this entire circuit behaves like a quantum object. And these kinds of man-made quantum objects are really wonderful because they create new paradigms for communication. In fact, quantum communication protocols have been around for a long time. There are actually companies that have developed secure communication channels that are used by banks that use entangled states of light to encrypt communications between banks. But in the future, we'd like to develop new technologies, say for simulation or for quantum sensing or quantum computing. And that's really what I want to highlight a little bit. But it is these advances in our ability to control the quantum world that have allowed us to go from fundamental physics to hopefully future quantum technologies someday. Okay, now I want to highlight just some of the cool things, just to get a sense of what is it like to be a quantum experimental physicist. So I mentioned, these quantum states exist, but it turns out they're pretty fragile. And so you have to come up with extreme conditions to realize them and make them stable. So for example, in my group, we have these large cooling apparatuses. This is called a dilution refrigerator. And we have to put quantum objects, the quantum systems that we study, the circuits, the devices, the chips that we study, we have to put deep inside of this kind of cooling apparatus that can get to extremely low temperatures. So temperatures that, to give you some sense, are around 10 millikelvin in temperature. So that in the Fahrenheit scale, that's about negative 460 degrees Fahrenheit. These are the coldest temperatures that you can imagine in the natural world. And in fact, they're some of the coldest temperatures that you can find anywhere in the known universe. So if I say took some object and I placed it in interstellar space and I let it sit there far away from other galaxies and light sources and planets and things, that object would cool down, but it would stop, and it would stop cooling at around 2.7 degrees Kelvin. So hundreds of times hotter than this. And the reason that it stops at that 2.7 degrees Kelvin is because there's, the microwave background radiation from the Big Bang continues to heat it up. And so the coldest places that we know of in the known world, in the known universe, are on Earth in quantum physics labs. And if you look inside of this thing, what we do is we put devices and things to measure them. And sometimes another extreme condition that we have to apply are very large magnetic fields. So we have to sometimes place these systems into some of the biggest fields that you can get easily. So like 14 Tesla, to give you some sense, some of you may have had like an MRI at the hospital. The big magnets that are used for those kinds of things are typically like 2 or 3 Tesla. So this is quite a bit bigger than that. Or to give you a sense, like the Earth's magnetic field is about 50 microtesla. And so this is many orders of magnitude larger than that. Now, because these quantum states are really fragile and we have to cool them down, it also means that if we want to do anything with them, like control them or measure them in these circuits and devices, what we have to do is we have to do ultrasensitive electrical measurements. And in my group, we do a lot of ultrasensitive microwave measurements. So we have to send very, very dilute microwave signals down into these large cooling apparatuses to measure the circuits and to figure out what they're doing, and to control the qubits. Now, I promised you I would tell you what a qubit is. I've said this word a couple times, and I just want to highlight what that actually means. So a qubit is it's shorthand for a quantum bit, and it is essentially the building block of a quantum computer. But actually, it turns out it's the building block for all these other things too, for like quantum sensors or quantum communications. You need qubits to be able to do anything. So a qubit is like the quantum analog of a classical bit, which is the simplest unit of information in a classical system. So actually, I have to credit my colleague Angela Wilson for this really wonderful analogy here, for trying to understand what a qubit is, again, in an imprecise classical language. So qubit states, quantum states, are often represented as zeros and ones, very much like classical bits are represented as zeros and ones, except we put this fancy bracket around it for fun. But imagine that you have a classical bit this can be any kind of system that can be in one of two states, so on or off, up or down, left or right, whatever you want, two distinct states that we represent as 0 and 1. And you could think about a light bulb as being on, that's the 1 state, and a light bulb being off, that's the 0 state. Well, in a classical system, those are the only two options you would have. But in a quantum state, what you actually have is the ability to combine on and off, 0 and 1, in an arbitrary superposition. So this is that superposition idea again. You can have these two states existing simultaneously. And so Angela has this great way of describing, like, here's, think about a dimmer switch in your house. You can continuously vary the light, not fully on, not fully off, but you get all of the values in between, or you have access to all of the values in between. And this is really at the heart of why these quantum technologies are powerful, because in order to describe a light bulb in a classical sense, you just need to know if it's on or off. But in a quantum sense, it turns out you have to know a lot more, and you have to know exponentially more information to understand qubits. And it's this exponentially larger amount of information that's needed that's really at the heart of what makes these technologies ultimately potentially so powerful. Here's just a bunch of pictures of, so that's in the abstract what a qubit is, but there's many different ways you can imagine making qubits. So like IBM and Google make these superconducting circuits. There are trapped ion systems. There's a company called IonQ, which is about to go public, honestly, to they use trapped ions for quantum computing. You can use electronic defects inside of diamond or other crystals. You can do trapped electron systems. That's one of the things that my group works on. All these kinds of things. Okay, and so what do these things actually look like? So here's a quantum, so those are the qubits, those are the building blocks, and you got to put a whole bunch of them together to make a quantum computer. And so here's a picture of a quantum computer at IBM. It's inside of one of these cooling apparatuses. You can see it's not that much. It looks very similar to the ones that we have in our group. It's a little bit bigger, but it's essentially the same kind of idea. So you have to cool these things down to very low temperatures. If you look inside of this thing, what you find is something that looks like this. Also looks like what's inside of our system. So the quantum computing chip is down here at the bottom, and all this other stuff is really associated with sending in the control and readout signals and getting everything cold and well isolated from the universe, et cetera, et cetera. Ion trap quantum computers. So this company IonQ has something that looks a little bit different. It's still pretty intense and complex. So you have this large laser system because these ions, which are trapped on chips that look like this thing. So there's a little central region here where they can trap, many tens or hundreds of ions, and then shine laser beams at them to control them in this. So this is what kind of modern day quantum computers look like. I'll mention these only have, at most, many tens of functioning qubits. That's kind of the state of the art right now. And this is exciting because there's a variety of different problems that quantum computing can solve. That's what the potential is. And I'll just highlight two of them here. So quantum computers in principle are hopefully going to be very helpful in trying to understand the structure of large molecules, which are inherently quantum systems, or for the discovery of new materials. Inherently, these kinds of systems, which are currently solved, are very difficult problems for classical supercomputers to solve because they're quantum systems. So they're really, really super complex because they intrinsically have quantum mechanics in them. And so a quantum computer is hopefully going to be better at kind of doing those kinds of things in the future. There's also probably, you've heard that quantum computers are really good for difficult problems in encryption and information and cybersecurity. So there's protocols for developing ways of breaking even the best encryption right now using quantum computers. Now there's an important caveat here. There's a ton of hype surrounding this kind of stuff, but there's a lot of work that needs to be done. Actually, in order to get any of these kinds of problems to become realistically solvable on a quantum computer likely requires probably around 1 million or more functioning high quality qubits that all work together in just the right ways that we have perfect control over. Right now, the best state of the art is like 50-ish. And so we're pretty far away from being able to do these kinds of things, but this pace is accelerating rapidly. And that's actually an exciting place to be, because it means that there's a lot of jobs that are a lot of fun to do in quantum physics. So for example, I'm just highlighting a few that my group is interested in because they're kind of associated with experiment, but we have to make the quantum systems more resilient to noise and to disturbances. We have to figure out how to make them bigger. We have to figure out how to control them better, and we have to figure out how to interconnect them with each other and with different kind of, say, quantum memories or communication channels and all this stuff. But it's exciting. It's like the best time to be working in a quantum lab, and this is what the folks in my group do. This is just my quantum workforce dad joke on this. Okay, so now in just the little bit of time I have remaining, I just want to really quickly give you the 10,000-foot view of the kinds of work that we're doing in my group on trying to solve those kinds of problems, to make new and better kinds of quantum technologies. So like I already mentioned, we work on trying to develop new quantum computing platforms based on trapped electron systems. I'll say a little more about those. We trap them on the surface, actually, of a superfluid substrate at very, very low temperatures. We have a big program working in collaboration with other folks here at MSU to develop next generation quantum materials and to understand their properties. So materials discovery is a real driver. If you can come up with better materials with better properties, that's often it's historically been a fantastic way to push forward technological advances. We work with superconducting circuit-based qubits like IBM and Google does. And very recently, we've started working in collaboration with MSUQ researchers on these electronic defects in diamond. So again, I'm going to give you just a high level, elevator pitch on all of these kinds of things. And if you want to know more, you can check out our website. You can read our publications, look at more photos, hybridquantumlab.com. Or if you're on Twitter, you can follow us at LHQS _MSU. Okay, so here we go. Trapped electrons. This is the picture that I want you to have in mind. Electron floating on the surface of superfluid helium. This electron at very low temperatures. So this electron is actually shielded from other kinds of noise and decoherence and all kinds of bad stuff that's on these substrates by this very clean superfluid substrate on which it gets trapped. So we can place electrons deterministically on here and then use electrodes that are on the bottom here to control the quantum properties of these electrons. Now, the neat thing about this system is that it kind of brings together the best of both worlds from, say, ion trap systems and superconducting qubits. And we think that it's a much earlier and less mature field, but it's one that we have a ton of expertise in, and one that we think will ultimately be competitive with the best kind of quantum computing platforms that anybody can invent. And just to give you some sense, this is just, it turns out that these electrons, I've drawn them as little blue spheres, but that's just a cartoon. In reality, we have to have abstract ways of thinking about the quantum states of these electrons. So, but the electrons themselves, both their motion and their intrinsic magnetism, their spin, can be quantum states, and we can utilize those for quantum computing. And there's a variety of groups around the world. We're not the only ones working on this system, but it's an exciting time to try to develop. It's not every day you get to invent a new qubit, and that's what we're trying to do. Like I said, we work with superconducting qubits. We place them inside of these microwave resonator boxes where we can very delicately introduce single microwave signals in one port, control the qubit, and out the other port. And on these chips are little nanofabricated nanofabricated structures that actually have the qubits. These are made of superconducting materials. I show this just because one of the things that we do a lot of is a lot of nanofab. So we have to go in and actually engineer and design the devices that are then cooled down to low temperatures and studied. And then when we do, we can study things like superposition, or we can use the qubit to sense individual microwave photons that come in and out of this box. These are real data that we take in the lab. It's of the quantum properties of these things. And with these, we're not trying to compete with Google or IBM, but we're trying to learn about how to control these systems better, how to make them more resilient. Yeah, that's the kind of effort on the superconducting qubit front. And then very recently, we've started working with these electronics qubits in diamond. And these are very different. So you take a diamond crystal, and it turns out that if you zoom in, you get a picture of the diamond lattice. These green atoms are the carbon atoms in the lattice. And you can make a qubit pretty simply by just plucking two of the green atoms out, replacing one of them with a nitrogen atom, and the other one is left empty. That's what this V means. It means vacancy. So just imagine there's no atom there. And then this nitrogen atom coupled to this empty space actually forms an electronic structure that can be a qubit at room temperature even. So what we have to do is we can use microwaves to control the state of that qubit, but we've set up laser systems to shine. If you shine green light, you can initialize and read out the electronic state of these kinds of electronic qubits in diamond. Here's a picture of a piece of diamond that actually has a whole ton of these qubits in it. So we shine green laser light, and then the thing just emits red photons. And those red photons, if we collect them, give us the information about the underlying states of the qubits. And this is a big collaboration. This really involves a bunch of people from MSUQ. MSU has a real rich history in studying diamond devices and crystals in collaboration with the Fraunhofer Center for Diamond Coatings and Technologies here at MSU. And we've recently been working a lot with Sandia National Lab to develop new quantum sensor and quantum memory technologies with these. Okay, so I see that I'm a little bit over time, so I apologize for that, and I thank you for your patience. But here are the key takeaways. I hope I've convinced you that the quantum world is strange and counterintuitive, but you shouldn't be worried about that. It really is just, in some sense, a mismatch of language with intuition. We use our classical language. The quantum world just doesn't adhere to that kind of thing. But those counterintuitive properties are actually pretty powerful and can create awesome quantum technologies. And I tried to hopefully give you a little bit of a flavor of what specifically we do in the lab every day. And when I say we, it's not often me. I try to get down there as much as possible, but these are actually the wonderful postdocs, graduate students, and undergraduates here at MSU who do the heavy lifting to perform these kinds of quantum experiments. And it just simply wouldn't be possible without them. And finally, it's highly collaborative work. You can imagine it brings together a bunch of different disciplines. I don't know how to do all of these things, so I have to rely on just a fantastic group of collaborators that do bring a ton of things to the table that I simply don't know anything about. And also all the folks that really help to push the funding of this work. It's building a quantum computer is not cheap, but it will hopefully have amazing impacts for the world.